Hello everyone and welcome to the new session of the lecture series, and today's topic is plant growth and development. So we have already discussed in the previous lecture, we were discussing about some of the terms such as plasticity, and then we studied the conditions responsible for development, and then we studied about the discovery of the phytohormones, which were auxin, gibberellin and cytokinin. And these hormones are all plant growth enhancer, or you can say plant growth promoting. All right. So again, I would like to mention all the topics covered in this lecture are from the NCERT book. All right. So now today we'll be studying about the rest of the hormones. First, we will study about the discovery of the hormones. Okay. So let me first give you a brief, background or a brief revision of the discovery of the phytohormones. So let's start with auxin first. Okay, let's do it quickly. Charles Darwin and his son Francis Darwin, he was monitoring the canary grass coleoptile, movement of these coleoptile towards the light. So what they concluded that there was a substance, that guide the canary grass go towards the light. All right, and later FW went identified that substance as auxin. And gibberellin, how was gibberellin discovered? So there was a foolish seedling disease, known as Bacne. This disease was caused in rice seedling by the fungus Gibberella fujikori. So later in Japan, E. Kurosawa, he was a Japanese. He reported what he did? He did an experiment in which he infected the uninfected rice seedling. Or you can say he treated the uninfected rice seedling with the sterile filtrate of the fungus Gibberella fujikori. And he reported the appearance of the symptoms. All right. And then later, the active substance was identified as gibberellic acid. So gibberellic acid was causing the growth in the rice seedling. That's why it is known as foolish seedling. There was increased growth of the internodal region, or you can say of the main axis, which was caused by gibberellic acid produced by the fungus Gibberella fujikori. Now, let's study about the discovery of cytokinin. So Skoog and colleagues, what they were doing an experiment on tobacco stem internode. And what they observed? They observed that there was callus proliferation only when auxins were supplied, and they were supplemented with specific nutrients obtained from either the extract from the vascular tissues or from the yeast extract, or from coconut milk, or from the DNA. Alright, so for callus formation, in addition to auxins, extract from these substances was also required. Alright, so what happened? These additional substances, they triggered the successful proliferation of the callus, and later Skoog and Miller identified and crystallized this substance, which promotes the cytokinesis, means cell division, and they named that as kinetin, which is a cytokinin. Now, today let's talk about the discovery of the phytohormone in detail for abscisic acid. All right, thus we have already discussed. So, what happened during the mid-1960s? What happened? 3 independent researchers, means abscisic acid was identified 3 times. By 3 independent researchers. So what they reported? They reported the purification and the chemical characterization. They characterized also this substance, of 3 different kind of inhibitors. So abscisic acid we know is a plant growth inhibitor. It is a plant growth inhibitor. So 3 independent researchers, they purified and they characterized, mean structure, elucidation and all and everything, of an inhibitor. And what they named individually, they named one as inhibitor B, other one was named as abscission 2, and third one was named as dormin. So inhibitor, because it inhibits the plant growth, so they named it as inhibitor. Abscission. Abscisic acid also play a role in abscission. Okay, we'll talk about this in later slides. And then dormin. Okay, it induces dormancy in seeds and bud. Okay, that's why dormin. Okay, now all these inhibitors, all of these three, okay, which was identified individually by three independent researchers, they were all chemically later identified as abscisic acid. Okay, they were named abscisic acid later. So this was about the discovery of abscisic acid. Now let's study the discovery of the phytohormone ethylene. Okay, so ethylene, Cousins, okay, they confirmed the release of the volatile substance. Okay, ethylene, we know that it is a volatile hormone. It is a volatile hormone, or you can also say it is present in gaseous condition. It's a gaseous hormone. All right. So Cousins, they confirmed the release of a volatile substance from ripe. This you have to remember, ripe organs. Okay, and this volatile substance accelerated the ripening process. Okay, of stored unripe bananas. Okay, so they help the unripe bananas to ripe. Okay, means it helps in ripening process. So that's why ethylene is also sometimes known as the ripening hormone. Okay, now, so later this volatile substance was identified as ethylene. Okay, a gaseous plant growth regulator. Okay, or phytohormone or plant hormone. Okay, now let's study the function of these phytohormone. Alright, so let's start with auxins first. Okay, let me introduce you, okay, again to what is auxin, okay, and how its name was derived. So auxins derived from the Greek word auxin. Okay, auxins, okay, is the word derived from the Greek word auxine, okay, which means to grow. Okay, and auxin was initially, okay, extracted from the human urine. Okay, so the first auxin extracted was from the human urine. All right. So now where is auxin synthesized? Auxin is primarily synthesized in the growing regions. Okay, what are the growing regions? You can say tip of stem and the tip of roots, or you can say meristem, shoot apical meristem, or okay, root apical meristem. All right. So auxins are usually produced in the growing regions. And what does growing region contains? They contain meristematic cells, which are undifferentiated cells. All right. Now auxins can be either natural auxins or they can be synthetic auxins. Examples of natural auxins are indole acetic acid, okay, and another one is indole butyric acid. Okay, why they are auxins are indole derivatives? Okay, means they contain an indole group as the parent nucleus. Okay, now talking about the synthetic auxins, examples are NAA, naphthalene acetic acid, and 2,4-D. Okay, what is 2,4-D? 2,4-dichlorophenoxyacetic acid. Okay, this is also an auxin, synthetic auxin, which is used as a herbicide. Now, what is a herbicide? That kills, you can say, herbs or weeds. Okay, it kills the weeds. Or you can, and most of the herbs and weeds are dicots. So you can also say it kills dicot, but it has very limited or no effect on the monocot plants. Okay, but it is in high quantity toxic to the dicots such as weeds. Alright, so that's why it is used as a herbicide. Now let's talk about the functions of auxin. Okay, what is the primary function? Primary function is the initiation of the root development. Okay, so if you add auxin, okay, in the suppose callus, okay, then it will lead to the development of root. Actually, a ratio of auxin to cytokinin is required, okay, but auxin is more important for root development. Another function is it promotes flowering. Okay, so means what it does, initially the plant is in vegetative phase. It helps in the transition of plant from vegetative phase to reproductive phase. Okay, flowering is a reproductive organ. Alright, and where flowers are produced, suppose they are produced at the shoot apical meristem. Okay, shoot apical meristem modified to produce flowers. Alright, so auxin is important for promoting flowering. Another role is it prevents the premature shedding of young fruits and leaf. Alright, so it prevents the falling off, or you can say shedding means falling off. Falling off of young fruits and leaves, but it promotes the abscission of mature leaf. So if we talk about mature leaves, it promotes the shedding, or it promotes the abscission of the mature leaf and older fruits. Okay, now what is abscission? Abscission is the, you can say, falling off of the plant organs. Okay, such as, in the bracket you can write leaves, it can be fruit, okay, a ripe fruit or a mature fruit, or a flower. Okay, so this is known as abscission. So let me explain you abscission a little bit. So if this is a leaf attached to the stem, alright, if this is a leaf attached to a stem, abscission layer is formed here. Okay, in this region, abscission layer will be formed. And why abscission layer is formed? Okay, there are a lot of hormones involved. Major hormone is auxin. Alright, cytokinin and ethylene are also involved. Okay, but major is auxin. So here an abscission layer is formed. This is known as the abscission layer. So due to this formation of abscission layer, what happens? Leaf is detached from the main axis and it falls on the ground. Alright, so this is known as abscission. Another important role of auxin is it promotes apical dominance. You have to remember this. Auxin promotes apical dominance. Now what is apical dominance? Apical dominance is when the apical bud inhibits the growth of the, or suppresses, you can say inhibit or suppress the lateral bud. This is known as apical dominance, which is caused by the production of the auxin. Okay, this is caused by the production of the auxin in where? In apical buds. or you can say shoot apical meristem. All right. So this is clear. So now another term is decapitation. So what we do in decapitation? There is a removal of the shoot tips. We remove the shoot tips where auxins are produced. Okay, auxins are produced here. Okay, shoot tips in the growing region of the shoot tips. And what happens? Due to the removal of the shoot tips, no auxin is produced. And what happens? It results in the growth of the lateral buds. All right. So this technique is widely applied in tea plantations and hedge making. Okay, when we have to go for the lateral branching. Okay, when we want to initiate the lateral branching or promote the lateral branching, then we do the process of decapitation. Alright, so in this figure you can see apical dominance in plant. Figure A, a plant with apical bud intact. Okay, so this is a apical bud. This is the apical bud enlarged. Okay, or you can also see region of shoot apical meristem. Alright, now here in the figure B, a plant is shown with apical bud removed. Alright, so here you can see there is no lateral growth. But whereas here you can see emergence of the branch, lateral branch, there is lateral growth is seen here due to the removal of the apical bud. Alright, so this is clear. Now, another important function of auxin is it induces parthenocarpy in fruits such as tomatoes. Alright, now what is parthenocarpy? Parthenocarpy is the development or formation of fruit, okay, without fertilization. Okay, so without fertilization, fruits are developed. Okay, and this phenomena is known as parthenocarpy. Okay, and what does it do? As there is no fertilization, so sterile fruits are produced. Okay, and what are sterile fruits? Means they don't have seeds. They have no seeds. Okay, so this technique is applied, okay, in tomatoes and we get seedless tomatoes. Okay, another example can be watermelon. We get seedless watermelon also. Okay, now next function of auxin is the herbicidal activity. This I've already explained. Auxins have the herbicidal activity, means they can act as herbicide. Okay, example is 2,4-dichlorophenoxylic acid, 2,4-D. Okay, so auxins usually they kill the dicotyledonous weed without affecting the monocotyledonous plant. Okay, so because of this characteristic of auxin, it is used to prepare weed-free lawns by gardeners. All right, so auxin has a commercial importance here. Okay, so 2,4-D, okay, sold in the market and it is used for the preparation of weed-free lawns by the gardeners. All right, now coming to the next function, auxins, they control the xylem differentiation. Okay, xylem differentiation means initially the cells were undifferentiated, they will undergo differentiation. Okay, and auxin is involved in differentiation and they will form mature cells. So usually the xylem differentiate into 4 types of cell. We have xylem parenchyma. Okay, xylem consists of 4 types of specialized cells. Then we have xylem fibers. Okay, then we have vessels and we have tracheids. So xylem help in So auxin helps in xylem differentiation. Another role is it helps in cell division, and you can say it also helps in cell elongation. Okay, it helps in cell division, and elongation means it helps in cell growth. Okay. Now let's talk about the next hormone, which is gibberellin. So more than 100 gibberellins has been discovered. Okay, till now. Okay, and they are denoted as like gibberellic acid 1, 2, and 3, and so on. Okay, till you can say gibberellic acid 100. Okay. Now, first gibberellins which were discovered was GA3. It was named as GA3. So, gibberellins, also known as gibberellic acid. Why they are known as gibberellic acid? Okay, because they contain an acidic group. They are acidic in nature. So, it is also called as gibberellic acid. All gibberellic acids are acidic. Alright, now let's study the functions of gibberellins. So first major function is the increase in length of the axis. Okay, or you can say increase in the length of the stem. Alright. Example can be grape stalk, increase in the length of the grape stalk. Okay, you remember that Gibberella fusiculae, which secretes gibberellic acid, it causes the enhanced internode growth. Okay, similarly, if we apply exogenous gibberellic acid, then there is an increase in the length of the axis. One example I have given of the sugarcane. In what sugarcane, what happens? The increase in the, so this also have a commercial importance here. So increase in the length of the stem, okay, increases the yield in the sugarcane as much as by 20 tons per acre. Okay, so this is the commercial importance of gibberellic acid. Alright. Now next is another important function of gibberellins is delay in the senescence. Okay, it delays senescence. So now let's talk about senescence. What is senescence? Senescence is also known as biological aging. Okay, it is also known as biological aging. And what happens during senescence? The morphological as well as the, you can say, morphological characteristic of the organism or the cell and the function of the cell or the organism deteriorate. Okay, they start to deteriorate. Okay, ultimately this leads to the lack of organization and function of the organism. Okay, and senescence finally leads to death, lack of organization and function in organism. Alright, and senescence, which is inevitable, will finally lead to the death of the organism. Alright, so gibberellins, okay, they delay senescence. Alright, so what happens during senescence? A lot of things are happening. First is oxidative damage. Alright, then we have the chlorophyll degradation. Chlorophyll degradation, disintegration. Another thing happening is photosynthetic apparatus disintegration. Photosynthetic means there will be no photosynthesis if so photosynthetic apparatus is deteriorated. So basically, gibberellic acid as well as cytokinin also, we'll study about cytokinin also. Cytokinin is the major hormone that delays senescence. Gibberellic acid also delays senescence. Okay, so they reverse these changes. These changes happen during the senescence. Alright. The next function of gibberellic acid is to speed up the malting process in brewing industry. Okay, so malting process is very important in the brewing industry. And what is malting? It is the sprouting or the germination of the seed, or germinating seed, germination of seeds in, you can say, water or in moist condition. Alright, so malting involves first soaking. Okay, first is soaking of the seeds in water. Second is the germination or sprouting of the seed. Alright, now next function of gibberellin is gibberellin causes fruit like apple to elongate and improve its shape. Alright. Now, next function of gibberellin is it hastens the maturity period in conifers, thus leading to early seed production. Okay. So, it hastens the maturity period, means it helps the plant to reach the reproductive stage early. Okay. It helps the plant to achieve the reproductive stage. Alright. And once the reproductive stage is achieved, what happens? Early seed production can occur. Okay. So this is clear. Now coming to the next function, gibberellins, they promote the bolting. Okay, bolting process. What is bolting? Bolting is the internode elongation just prior to flowering. Okay, so this is very important, and this can be seen in beet, cabbage, and many plants with the rosette habit. Okay, with the rosette habit. So gibberellins promotes the bolting process, means internode elongation just prior to flowering. Okay, so this was about gibberellins. Now let's talk about cytokinin. So cytokinin, okay, let's study about the brief introduction about cytokinin first. So cytokinin was first discovered from the herring sperm DNA. Alright. This is very important to remember. It was discovered from the herring sperm DNA. Alright. And later zeatin. So kinetin is a synthetic hormone. Kinetin is a synthetic hormone. And later zeatin, which is a natural hormone. Okay. This was discovered, okay, it is a natural hormone. This was discovered from the corn kernels and coconut milk. Okay. Now cytokinin, okay, light auxins, they are synthesized in the growing root regions, okay, such as the root apices, root apical meristem, developing shoot bud, and young fruits. Okay. And what does cytokinin do? It helps to produce new leaves, chloroplast, okay, in the leaves. Okay. So it helps to produce new leaves as well as chloroplast in the leaves. Now let's come to the function of the cytokinin. So lateral shoot growth and adventitious root formation is promoted by auxin. Okay. So there is a promotion of or enhancement of the lateral shoot growth and adventitious root formation if we applied cytokinin exogenously. Okay. And what is adventitious root formation? Formation of the shoots, okay, from a region other than the natural region, means other than the usually shoots are found from the nodes or branches are found from the nodes. You can say formation of shoots from internode instead of node, from the internodal region. Alright, this is known as adventitious shoot formation. Okay. Now, another role of cytokinin is overcome the apical dominance. Okay, apical dominance was when the apical bud suppresses the growth of the lateral bud. So, cytokinin do the reverse. Okay, it overcome the apical dominance and promote lateral growth. Okay. The next is major function of cytokinin is it delay the senescence. Okay, and how it delay the senescence? By promoting the nutrient mobilization. All right. So if this is a leaf, okay, and cytokinin is applied in this region. Here cytokinin is kinin is exogenously applied. So what happens? All the nutrient will be directed towards this region. Okay, all the nutrients are directed to the region where cytokinin is applied, and this region will remain healthy. It will remain green. Okay, there will be no deterioration of the chloroplast and chlorophyll. I've already told you during senescence what happens. Oxidative damage, photosynthetic apparatus degradation, chlorophyll degradation. Here, cytokinin will reverse all the effects. Okay, and it will delay the senescence. So this was the end of this lecture. We have discussed about the discovery and the function of the plant growth promoters. Okay, auxin, gibberellic and cytokinin. We will discuss about the plant growth inhibitor hormone, which is abscisic acid, and then we'll discuss about the ethylene and rest of the terms in the next lecture. Thank you very much for listening.